Neurons and the nervous system. Okay, so the nervous system is something that is unique to animals. It is going to allow us to almost instantly respond to stimuli. Okay, so for instance, if I put my hand on a hot stove, I can remove my hand from that hot stove, right? If I put a plant on a hot stove, it doesn't do anything, right? The plant just dies because it can't move that fast. It can't. It doesn't have muscles or nerves or anything to be able to respond to it. So this is a pretty important thing in the evolution of animals that allows us to respond to stimuli. Okay, and not only respond to it, but also respond to it very quickly. Okay, we can say that this is a system of cells. That use electrical impulses to transmit signals about environmental stimuli. Okay, so we divide up the nervous system into two main parts. Okay, the first part is called the central nervous system. Which is just abbreviated CNS. And there's two parts to the central nervous system, and those parts are the brain and the spine, or the spinal cord. Okay. These are the parts of the nervous system that are not super well understood, and we know that there's some like processing of information that goes on in our brain and our spinal cord, but we really don't have any idea how that works, right? So like this whole consciousness thing, we don't really know how consciousness functions. We don't know like why I can look at this and think about, oh, this is a green Nalgene that I purchased in high school, and where do all these memories come from? How are they stored in our brain? We don't really get it, right? So it's a little bit magic-y at this point, okay? Very similarly to probably the way that you guys think about computers for the most part. So if you think about a computer, what happens in that little box of the computer, that part is mostly magic, right? Let's be honest. So like if I click on Internet Explorer and I type in Chrome, I click on Chrome, right? And I type in my favorite website, it brings up that website from somewhere else on the planet. It's pretty magic, right? The brain and the spinal cord are doing all that processing that's doing the magic-y stuff. The rest of it, though, is called the peripheral nervous system. And to be honest, it's much easier to understand. We get how this works, okay? The peripheral nervous system is all the other stuff that's attached to the computer, right? It's the mouse, right? So you can input information into the computer using the mouse. It's the keyboard. You can input information that way. It's your webcam that's taking information about the room around it. It's less magic. We understand how it works. These things that are providing information to the central nervous system, we call sensory neurons. Then your brain and spinal cord, the central nervous system, can give you feedback on what you should do about those situations, right? So in the case where I put my hand on a hot stove, my brain gets the signal that it's hot from sensory neurons, and then it sends a signal back that says, move your hand. So I move my hand out of the way. These are called motor neurons. They're the things that are actually going to process the signals that are coming out of my brain. Okay, so in the case of the computer analogy, these are all your output signals. This is like your printer and your speakers and your monitor, right? Anything that's getting information from that box, the magic box, right? Then you've got the stuff that's in between. And the stuff that's in between is not really doing anything on its own. It's just relaying a signal, okay? And we call these interneurons. So the sensory neuron is actually, sending a signal, but the signal has to travel through these interneuron things to get to the brain and then travel back. Just like your mouse has a wire attached to it, the wire itself is not doing anything other than just sending the signal from the mouse to the computer, right? The wire is like the interneurons. We divide up the nervous system into two main cell types, okay? Two main cell types in the nervous system. Number one, the one that everybody should know about, the neurons. Neurons are super important. They're the actual thing that sends the chemical signals, or the electrical signals, rather, sorry. Okay, so these are cells that can send electrical impulses from their cell body to their axon terminal. Okay, we'll talk about those in just a minute. Okay, then you've got these other types of cells. These other types of cells are called glial cells. Sometimes you see them called neuroglial cells. Okay, and these are the rest of the cells, right? So basically, any cell that's in the nervous system that's not a neuron is called a glial cell. And there's a bunch of different types of them. We're going to focus on like maybe 3 or 4 different types, but there's a bunch, 20 plus different types of glial cells. Okay. Then let's focus then on the most important type of cell, the neurons. Okay. So neurons are salty bananas. We talked about this before in this class, but it is helpful, I think, to think of neurons as salty bananas. So if you imagine opening up a banana and putting it on a plate and then putting some salt on it, that gives you a good indication of what a neuron is, right? So on the outside, we've put salt, right, which sodium. I want you to make that connection between salt and sodium. And that'll tell us that on the outside of a neuron, you should expect to see salt or sodium ions. Okay, so Na plus ions are outside the neuron. But inside the neuron, the banana part, we know that bananas are a good source of potassium, and so we can remember that K plus ions are on the inside. Now, the amount of sodium on the outside is greater than the amount of potassium on the inside. This is due to the way that the sodium-potassium pump functions. And therefore, the neurons are always going to end up with a net negative charge. And that net negative charge is going to be roughly negative 70 millivolts. Okay? This net negative charge is something that is referred to as resting potential. And the resting potential of negative 70 millivolts is maintained by that sodium-potassium pump, and make sure that it stays around negative 70 millivolts by pumping the sodium ions out and potassium ions in. Okay? One thing that you might not realize about electricity and about voltage is that there's really no such thing as an absolute voltage. Okay? Voltage is always based on the difference between two areas. Okay, so for instance, in this case, this negative 70 millivolts means that there is a 70 millivolts difference between the outside of the neuron and the inside of the neuron. But if you raised the voltage on the outside, right, that would make this difference greater. If you lowered the voltage on the outside, that would make this difference less, right? Even though we're not actually changing, thank you, we're not actually changing the voltage of the neuron itself. By changing the voltage outside, that changes the relative voltage of the neuron, okay? So if you've done like AP chemistry, you know about like differences in electrical potential of cells, of the galvanic cells, right? Or if you've done physics, you know that if you take something that's 12 volts and you ground it to something that's 5 volts, that the overall voltage running through it will be 7 volts because it's the difference between the two, right? These are just basic electrical things, right? So, neurons function by sending an electrical signal from their dendrites to their axon terminal. Okay, and so I used some words. So let's define these words and figure out what they are. I'm going to draw a neuron here. Okay, so we got some parts to this neuron. You might be familiar with it already. This is called the cell body here. Occasionally you might see the cell body referred to as the soma. I'm not sure if anybody actually uses that term anymore, but sometimes you might see it. Okay, you've got this long space here. This is called the axon. Okay, the end of the axon is called the axon terminal. And then over here, things, little spread out parts on the cell body are referred to as dendrites. Okay, so they get a signal on their dendrites here. That signal causes the cell body to depolarize. That sends the signal down the axon to the axon terminal, and then the axon terminal sends the signal to the next neuron. Right? Okay. So, let's label on here the signal direction. Say that we're going this way. Okay, so let's sort of map how this works. Here's the signal transmission in a neuron. So it all starts out when the dendrites receive a signal. Okay, so the dendrites are stimulated, and they can be stimulated by various things. They can be stimulated by a neurotransmitter released by the previous neuron, or if it was the case of a sensory neuron, they could stimulate themselves by having some chemical reaction happen around, like if it's in your eye, the presence or absence of a certain wavelength of light, or if it's like a touch sensor, the pressure around it can cause those dendrites to be stimulated. And when the dendrites get stimulated, that causes Ca2 plus ions to enter the cell body. Okay, having Ca2 plus ions enter the cell body will in turn depolarize it. Okay, or make it go from its resting potential, which is around negative 70 millivolts, up to another value. Okay, so the Ca2 plus ions raise the voltage of the cell body from negative 70 millivolts, which is resting potential, to negative 55 millivolts, which is called the threshold. Okay, so that threshold then causes, when it reaches negative 55 millivolts, is going to cause the gated ion channels, the sodium gated ion channels, to open up. So negative 55 millivolts is the point where the Na plus gated ion channels open. Okay, and then as a result of that, Na plus ions rush into the axon, opening more ion channels, and making the axon positively charged. This is called action potential. Action potential reaches the axon terminal. And the axon terminal releases neurotransmitters and those neurotransmitters then stimulate the next neuron. Stimulates the dendrites in the next neuron. Okay, and this is overall the entire function of the neuron, right, from start to finish. Now, the last thing that has to happen then, this thing is send its signal on, and the last thing that has to happen now is those sodium potassium pumps have to go to work, and they've got to get that cell back from whatever voltage it's at now back to negative 70, so it can be at resting potential, so that it can actually do its job again, right? Okay, so let's talk about this process. You'll notice as the notes go on, my care to make my neurons look good goes down. Okay, here's one neuron. We'll do an identical neuron below it. Whoa, what? Sure. Okay. So, as I said, the dendrites get stimulated, Ca2 plus ions from the synapse enter into the cell body. That depolarizes the cell body, which causes gated ion channels at this junction here between the axon and the cell body. It's got a name. Axon hillock, time. Causes all these gated ion channels to open like so. Okay, all the way down here. So Na plus ions are traveling into the axon like that. Okay, going down into here, and that's the action potential traveling down here, and then that causes the release of the neurotransmitters from the axon terminal. Okay? Now, the interesting thing, though, is that this process is actually pretty slow. Every time a gated ion channel has to open, that takes time. And, Amelia? These? It doesn't say anything. They're just little circles. They're like arrows and circles. No. Just circles. Okay. So every time a gated ion channel has to open here, that takes time. And so it's actually the much slower, or the slowest part about this whole process of having action potential travel down here. So, evolution has a very unique solution to this, and the solution is given to us by these types of cells that are glial cells called Schwann cells. Okay, and when you're born, you've got some neurons, most of your neurons, that have these very tiny little cells embedded on the axon of your neurons. Okay, and they just look just like this. And then as you get older and older and you develop, what happens is that these Schwann cells are going to secrete this protein called myelin. And the myelin is going to surround the axon like this. Okay? And the result of this is that every spot that is on the axon here, that's underneath a myelin bead here, is not going to be able to open up those gated ion channels because they're going to be blocked. Okay? So this means in this neuron here, you've got Ca2 plus that enters the cell body just like normal, depolarizes it. Okay? That causes Na plus to enter here. Okay, but now the next gated ion channel can't open because it is being blocked by this myelin sheath. And so it travels, that impulse travels down through the axon, opens the next one. That travels down through here and opens the next one. That travels down through here and opens the next one. And that travels down through here. Okay, the result of this is a massive reduction in the amount of gated ion channels that needs to open to have these to have this action potential travel down the axon. Okay, so let's say, just for the sake of ease, that this has 40 gates that have to open. But this one has 4 gates that open. Okay, the easy calculation then says that if this is the slowest part of this, that we can expect this to be roughly 10 times faster in a myelinated neuron than in a non-myelinated neuron. Okay? And this is one of those things that I love to look at and say, man, evolution, you crazy. Because this is like the least elegant solution to this, right? So this is what initially evolved, was this neuron that had all these gated ion channels on it, right? But that was pretty slow. And so this one, instead of just having less gated ion channels, which would solve the same problem, it would do the exact same thing. Instead, there's an additional cell that secretes an additional product, myelin, that covers up most of the gated ion channels, which then makes it more efficient. Okay, so this is definitely not the most elegant way of solving this problem, but it's the way that evolution solved it, and so that's what we have. Right? So, like, it's actually really stupid that I hear a lot of times when people learn about the myelin sheath, they talk about like electrical wiring. Like, look at your extension cord; it's got a plastic sheeting on the outside, and that stops it from, you know, shorting out, and it makes it insulated. That's not what the myelin sheath is about at all. The myelin sheath is about destroying the functionality of the neuron, but it happens to be that if you destroy the functionality just enough, it actually gets better, right? So you limit the amount of gated ion channels that open, and therefore you speed up the rate of transmission, right? Okay, so this is the myelin sheath. And the myelin sheath is a substance secreted by a glial cell called a Schwann cell. That increases the rate of transmission in a neuron by blocking some gated ion channels. Okay, so we've talked about the role of the neuron. We pretty much, I hope at this point, understand how the signal travels down a neuron. Okay, now I want to talk about how the signal travels between neurons. Okay, so if you have a neuron here, and then a neuron right next to it, like this, okay, the space in between is referred to as the synapse, right? So if we sort of took this space in between and we blew it up, something like this, okay, now you've got your axon terminal, okay, and then you've got the cell body over here, and then you've got the dendrites. Sure, something like that. Okay, so this is the axon. Terminal. And these are the dendrites of the next one. Dendrites and cell body. So once this signal travels down to the axon terminal here, the axon terminal is going to get stimulated and will release these things called neurotransmitters. Okay? These neurotransmitters are molecules that can be absorbed by the dendrites of the next neuron, right? So they might go into this spot here, into that spot here, into this spot here, and into this spot here. And if enough of these neurotransmitters are then absorbed by the next neuron, that neuron will then have Ca2 plus ions enter into the cell body, and then that will depolarize the cell body and send the signal down the axon. Yeah? Yep, there's always Ca2 plus ions in the synapse. In fact, Ca2 plus ions in the synapse are one of the things that contributes to the negative 70 millivolts resting potential anyways in the first place. Okay? So, this thing here, like I said, is called the synapse. And the synapse is just the space in between two neurons. Sometimes there is a synaptic vessel, meaning like a space that's containing just a couple of neurons, maybe like 3 or 4 neurons. And sometimes there's just like a bunch of neurons, hundreds of neurons that are all sort of emptying out into the same synapse. Okay? So, we divide neurons up, or we divide synapses up into 2 different types of synapses. Okay? The first one is called a chemical synapse. So chemical synapses are going to rely on these things called neurotransmitters. Chemicals called neurotransmitters. Are released from the axon terminal and absorbed by the dendrites of the next neuron. This is by far the most common type of synapse in the peripheral nervous system. And there are also some pretty cool interactions that can happen here with other neurotransmitters that are released by the body by different systems. Okay, there are what are called inhibitory neurotransmitters. Like dopamine. Okay, and there are excitatory neurotransmitters. Like epinephrine. Okay. So, what do I mean by this? So let's say that you are laying in bed. Well, first of all, let's talk about what would happen normally. So let's say in this synapse here, in order for this dendrite, or in order for this neuron here to be stimulated to fire its action potential, it has to absorb 4 neurotransmitter molecules. That's a very low amount, but let's just say for the purpose of simplicity, that it has to absorb 4 of them. Okay? So if this thing doesn't release 4, then this thing doesn't fire, right? Okay. So let's say that you're laying in bed and you hear a noise. Right? It's the middle of the night. You hear like a crashing sound. You're like, somebody's broken into the house. I'm pretty sure they're here, and they're here specifically to kill me. Like I'm almost positive about it, right? And so you're laying in bed and you're freaked out now. You're wide awake and you can hear everything. You can hear the clock ticking on the wall. You can hear the air conditioning rattling around. Every time you move in the sheets, you can hear your skin like sliding up against the sheets. You know everything, okay? Why does that happen? Yeah, from epinephrine, right? So your body releases adrenaline or epinephrine, right? And what it does is instead of this neuron over here needing all 4 of these things to actually attach, 4 things that are released from here, there's a generic neurotransmitter, epinephrine, that goes in and attaches to 3 of the spots. Okay? It's a systematic. It goes throughout your entire body. And basically what it does is it makes it so now your neurons don't need nearly as much signal from the previous neuron to fire. Okay? So now if this neuron releases just 1 of these and 1 binds, that signal gets sent on. Whereas before it needed all 4 to get sent on. Right? And so that means that your body is going to respond to every little stimulus that it comes in contact with, right? Because you want to be in that state of hyper-awareness, okay? I don't know what it says about the human race or happiness or anything like that, but dopamine, which is the hormone that gets released when you're happy, makes it so you don't care about the world. it makes it so it's much harder to send a signal on. Every stimulus that you get, you don't really pay attention to. And the reason for that is that dopamine works by being an inhibitory neurotransmitter. Meaning instead of acting like the epinephrine where it's filling up the spots here and causing it to only need one neurotransmitter to then activate, dopamine goes in and it actually binds to these sites, but it doesn't activate them. So it just blocks them. So it makes it so this neuron here has a significantly less chance or lower chance to fire because now it has to find a place for these 4 neurotransmitters to bind to when a lot of them are being blocked by dopamine. Okay? Yeah?
When would an example of dopamine be used? Like, you used adrenaline for being scared, but like
when you're happy. When you're just really happy, you look on the bright side, essentially. Like that term, looking on the bright side, just means like you're not paying attention. You just don't care about things. Now, you could use something that's similar to dopamine, right? So let's say somebody's really stoned, right? Same difference, right? They just don't care because they're not getting the signals. Like, you're super stoned and a car crashes behind you and you're like, right? Because you don't get that huge signal. Whereas somebody who's already stressed and freaked out, car crashes behind them, they're like, oh my God, I just had a heart attack, right? Because you get that signal much more, what's the word I'm looking for? Like the signal is a lot greater when it travels to your brain because you're getting it from a lot more of your neurons because it's taking less of that signal to get passed on, right? Okay, so now let's talk about electrical synapses. Electrical synapses work by changing the voltage of the synapse. By absorbing or releasing ions. Okay, so the 2 things that can happen in an electrical synapse are, first of all, this is by far the most common in the central nervous system. And there's 2 things that can happen. The first one is called hyperpolarization. Okay, so if the synapse gets hyperpolarization of the synapse, that means the synapse gets even more positive. So instead of resting potential being -70, maybe resting potential is now -150, right? Does that make it more or less likely for the neuron to fire? What does it have to get to fire? -55. So if normally we're at -70, we've only got to go, 15 to get to -55, right? But if we're at 150, -55 is 95 millivolts away. So that's very difficult to do. So the hyperpolarization of the synapse is going to be an inhibitory reaction. Okay, whereas depolarization of the synapse can be an excitatory response. Okay, meaning like, let's say instead of resting potential being -70, maybe resting potential is like -58. So now you only have to go 3 millivolts up. That's like a couple of Ca2+ ions entering, and then it'll fire. Okay? You can also depolarize it to the point where the next neuron fires. If the previous neuron's axon terminal absorbs enough Ca2+ ions from the synapse, that's enough to reach negative 55 millivolts in that cell body of the next one and cause that next one to fire without any ions entering. Yeah. Much more complicated. I think actually Monday's notes, but it's complicated. It's easier to explain how your body's natural pain resistance works. So like, let's say you get a cut on your finger or something like that, and like it really hurts right at the beginning, and then like, you know, over the course of a couple of hours, it's like still there, it still hurts, but like not nearly as much, and maybe it only hurts when you bump it on something. It's a process of hyperpolarization where essentially your body just gets sick of hearing about your thumb hurting, and so it stops responding to that signal, and so it says, okay, I get that our thumb hurts a little bit, and it hyperpolarizes to the point where it's like, only let me know if my thumb hurts if it really hurts, right? And so if it's able to hyperpolarize it so that it gets enough signal so that it gets from, you know, negative 150 to 55, then something has probably happened that you need to be aware of. But normally, if it's just sending like a constant "my finger hurts" thing, then your body's not going to it's not going to be able to travel to your brain because of hyperpolarization of the synapse. Questions on that? All right, that's it.